It is a distinct pleasure to introduce to you Professor Marc Kirschner, one of the leading authorities on how molecular processes are integrated into a biological functioning unit. Professor Kirschner graduated from Northwestern University in 1966 and received his PhD from the University of California, Berkeley in 1971. Following a postdoctoral research at Berkeley and at the University of Oxford, he was appointed an assistant professor in Princeton in 1972. In 1993, Dr. Kirschner arrived at Harvard Medical School from the University of California, San Francisco, where he had served on the faculty as professor for 15 years. At Harvard, he chaired the Department of Cell Biology for 10 years before founding the Systems Biology Department. Throughout his career, Professor Kirschner has attacked fundamental problems in cell and developmental biology, such as the dynamics and function of the cytoskeleton, the regulation of eukaryotic cell cycle, and the processes of signaling in embryos. He is a master of exploiting approaches of molecular and cellular biology and biochemistry to investigate the reactions and processes that exert spatial and temporal control over cells and organisms. Many of his findings and ideas were summarized in two books that he wrote with John Gerhardt, Cells, Embryos, and Evolution, published by Blackwell in 1997, and The Plausibility of Life, Resolving Darwin's Dilemma, published by Yale in 2005. This book can be found outside, and as somebody who published 2 books with Yale, I know that each time you buy a book, you earn a dime, so go and buy them. Professor Kirschner won many prizes for his remarkable achievements. He was elected foreign member of the Royal Society of London and is a foreign member of the Academia Europaea in 1999. He was the 2001 recipient of the William Rose Award presented by the American Society for Biochemistry and Molecular Biology. Later that year, he received the 2001 International Award of the Gardner Foundation in Toronto. And in December 2003, he received the Wilson Medal, the American Society of Cell Biology's highest scientific honor, named for an early 20th century pioneer, named after the early 20th century pioneer of American biology. This award is given to scientists who have made significant and far-reaching contributions to cell biology over the course of their career. Well, I have reliable sources in the Harvard Gazette, and I found the following story about Professor Kirschner. His first research job after college was working for Tony Gillette Corporation on hair straighteners. And he said in an interview to the Harvard Gazette, "I actually learned a lot about protein chemistry," he said, "and I avidly read everything I could in the National Journal of Sheep and Wool Science." The company asked him to stay on, but Kirschner elected to pursue a PhD. And he admitted, "I will never know what I could have accomplished in the hair products industry," he said. "But the truth is, I never regretted my decision to pursue fundamental biology." Professor Kirschner, please.
Well, thank you very much, Professor Levine. And I know a little bit about you from yesterday, and he's a specialist in sleep research. So I think as we look over the audience and everybody dozes off, I will think of your great contributions in this area. I also want to thank the organizers of this wonderful symposium. The subject matter is appropriate, of course, and the talks are wonderful, and there's, of course, more of an emphasis on people from Israel, which is terrific for me because I get to hear things that I don't get to hear so often, and I think every talk has been very informative and interesting. So this talk that I'm going to give is a little different in that it's more conceptual. I won't really be presenting any data, at least no data of my own. And it's an attempt to approach evolution from the point of view of what we've learned in the last 50 years from cell and developmental biology. So it's appropriate to this Evo-Devo orientation in developmental, on the issue of evolutionary biology. The quotation, having written 2 books on the general subject of the cellular and developmental basis of evolution, the one quote from Darwin that I remember precisely was, it's in his autobiography when he's talking about writing The Origin of Species, and he sort of kind of groans and says, "This book is taking over my life." So many of you maybe have felt that in the past as your work was just grinding you down. So this subject I want to talk about is called facilitated variation, and I will try to define it, explain it in the course of this talk. But it really comes from really the question of where phenotypic variation comes from, how it's produced, whether it's easy or hard to achieve. Now, this is a drawing, a series of drawings by Ernst Haeckel, who was an outstanding artist. Some people say that his art was more important to him than his science, but he was also a very important proponent of evolution, had translated Darwin's book into German, was very early in bringing Darwin's ideas to the continent. And here are pictures of faces of bats. They're very attractive, I mean, I'm sure, to other bats of the same species. And it illustrates just what tremendous, extraordinary variation in the phenotype exists in biology, amongst a small group of organisms. And the issue of phenotypic variation has not been dealt with as much, for good reason, as the issue of genotypic variation. And so I want to really focus on the phenotype, because in fact, it is the phenotype which is selected and not the genotype which is selected. The genotype only gets selected by virtue of being carried along during phenotypic selection. Now, traditionally, evolutionary biologists had little to say about phenotypic variation as compared to genotypic variation. And that's partly because at the time the field was coming together, there was very little that was known about how the phenotype was generated. One might say the first real clear experiment on the generation of the phenotype was the one gene, one enzyme hypothesis of Beadle and Tatum, who showed, in fact, that the phenotype, which was, in fact, a protein, an enzyme, was the product of a single gene. Now, that concept itself needs a lot of modernization, but it was the first early attempts, and of course, molecular biology and the whole DNA explosion in the 50s and 60s and up through now has led a lot of information to that. But even though that's told us a lot about how proteins are generated from genes, the phenotype is something yet much more complex. Now, some evolutionary biologists seem satisfied with only describing genotypic variation and selection. But if we only concentrate on genotypic variation and selection, we would never be able to explain these kinds of things that we want to explain in biology. That is, how was the eye created? How do we get this wonderful coloration and morphology? How about the human hand? And we wouldn't know how easy it was or difficult it was to achieve these forms of phenotypic expression. So my goal for this talk then is to focus on the means by which the phenotype is generated, and also to talk about what role the structure of the organism plays in the generation of the phenotype. And I'm going to start with defining evolvability, but after yesterday I have to put that in quotation marks. It's obviously a word which you know, is not considered something you can use in polite company. But I want to define evolvability. I want to show that the phenotypic variation of the type that we saw in those bat faces or the human hand or eye is facilitated by the phenotype itself. And I want to suggest some general principles based on reflecting on what we've learned in the past 50 years about cell biology and developmental biology for the principles of facilitated variation. And then I want to talk briefly about why or how this thing might have evolved in the first place. So this is an attempt, a different attempt than Dr. Evo used, but the same general purpose of breaking down Darwin's theory into sub-theories. A theory of natural selection, which Darwin got beautifully right. I mean, that was his great contribution, his great insight, and he brought to bear evidence from everything from breeding to the variation he saw in animals around the planet. He also knew he needed a theory of heredity because the variation that was being generated in each generation had to be carried on to the next generation. And Darwin also came up with a theory of heredity, which at the time was hailed as his second great accomplishment, which is now thankfully completely forgotten because it was wrong in all aspects. But ultimately, of course, a theory of heredity based on contemporaneous work, but unknown to Darwin, of Gregor Mendel led to an understanding of the concept of heredity. So we have today two good theories here, a theory of natural selection, a theory of heredity. They're not totally finished, but largely well understood. And the third theory we also need to have, and to make this complete, is a theory of generating the variation in the organism. That is the phenotypic variation in the organism. And that is the theory which has lagged behind these other two. Now, so one of the assumptions and I think accomplishments of understanding about genotypic variation is that genetic variation is random with respect to the environmental conditions in which it experiences. And people are, in fact, and we've heard a little bit of this today at this meeting, continually trying to chip away at that idea, and it may not, in fact, be completely true. But it's largely true if it isn't completely true. And the evidence is still not clear. There's still no clear evidence that it isn't completely true. But just because genetic variation is random does not mean that phenotypic variation is random. In fact, I'm not even sure what it means to say that phenotypic variation is random. And in fact, phenotypic variation cannot be random because it changes what already exists. What already exists is obviously not random, and therefore it builds on what already exists. So we know a little at this point that phenotypic variation cannot be thought of in the same terms as genotypic variation. Now, by 1940 was the general modern synthesis came about in evolutionary theory, and it was a major accomplishment of bringing together the conceptual advances, major ones in population genetics that Darwin, of course, didn't know about, with a sort of purification of Darwin's original ideas, removing the Lamarckian overtones that he subsequently added to his theory. Plus adding the experience from paleontology, but at that time in 1940, no one really knew how phenotypic variation was generated. But nevertheless, and this is the curious thing, despite their lack of knowledge, they actually had pretty strong ideas about what it had to be like. And Gould, in his Structure of Evolutionary Theory, summarizes what was the thinking about what phenotypic variation was like, and he thought that it had to meet three crucial requirements. That Gould likes to use big, complicated English words, so which, you know, even I have a dictionary to read his books. But anyway, so don't feel bad if you think you're an excellent speaker of English, you don't understand every word here. But anyways, three crucial requirements: that is, be copious in extent, that is, large amounts of phenotypic variation. Small in the range of departure from the mean, that is, very incremental at each interval. And isotropic, which meant essentially random, that is, going in all directions. And Gould argued that if it were not true that phenotypic variation had these properties, that selection could not otherwise operate as the creative force in evolution. Because if you imagine that there was very limited amount of phenotypic variation that was available, then that limitation would drive the adaptations that one saw in evolution. If the changes were large at each interval, then those big transitions would, in fact, be the ones that determine exactly the steps of evolution, rather than the small incremental selections that Darwin thought about. And if it were only focused in one direction and not in another direction, then you could say whatever was driving that direction was much more important than the selection that was occurring around it. So he argues that these three things were essential if selection would be the essentially only creative force in evolution. And I'm going to argue, in fact, selection, of course, is a tremendously important factor in evolution. That contribution of Darwin's is without doubt, but it's not the only force, and it's not the only determinant of the phenotype. Now, about the copiousness, that is the extent of variation, we come to the unexpected paradox of the conservation of genes and their diversity. The human genome is only about 1.5 times that of the fruit fly. 15% of human genes are shared with E. coli, 25% with yeast, 70% with frogs. So we're left with this question of there's a tremendous amount of evolution that's taken place, and yet there doesn't seem to be a correspondingly large amount of invention of genes. So at least on that level, there isn't as much variation underlying the phenotype as one might imagine. And although the tree of life that was just described as constantly branching is a good model for phylogeny, it isn't a very good description of what happened on the cellular and molecular level. What really happened are periods of invention and stasis, and between which there was a lot of regulatory evolutionary change. So about 4 billion years ago, we have the origin of life, and with that came the origin of metabolism, DNA, membranes, ribosomes, all the things that, and once that was invented, there was very little change in those mechanisms for the rest of time. So it's really it's sort of a picture of invention and stasis. About 2 billion years ago, and this is the subject matter that you take in a typical biochemistry course. So it seems a little stuffy and old. Much more recent and exciting, only 2 billion years ago, comes the material of a cell biology course that is the origin of the properties of eukaryotic cells, the nucleus, the cytoskeleton, the cyclin-driven cell cycle, signal transduction mechanisms, a variety of things, splicing, you can name them, that come up as seemingly as a suite of activities, but undoubtedly coming up if we had could look back in that time as being parceled together and then remaining stationary and unchanged. And this is why the study of yeast, for example, is so informative at the level of eukaryotic cell biology today for the study of human beings, because it's changed so little fundamentally. About 1 billion years ago came the origin of multicellularity. 600 million years ago, the origin of complex body plans. Of the 31 known phyla, 30 of them existed 530 million years ago in the Cambrian. For the last 530 million years, at most one phylum was invented. And it's sort of peculiar. Why is it, I mean, did somehow biology lose the capacity to make new phyla, or what? And I would argue that it didn't lose the capacity. It's just that the framework for innovation shifted. It was like wars, where the technology kept moving forward. And the old technology was still being used, but there was something new that was added to it, and all the innovation went there. So you can imagine that in the last 500 million years ago, there were lots of animals trying to generate new body plans, but they were probably lunch for all the animals that were making mouth parts and appendages, and where the actual innovation was occurring. So this is sort of the picture of limited amounts of evolutionary change in some areas, and then innovation in other areas. And this is not, in fact, the way that evolutionary biologists saw things. They thought that everything would change. Ernst Mayr made that very clear in his books, and that the only measure of the amount of change would be how long it had been since the common ancestor. But in fact, change occurs in some parts and not in other parts. Now, if these things are conserved, these core processes, that implies constraint to change, because there's no reason why mutation cannot occur in DNA polymerase or in ribosomes. But it means that all change in those things are lethal, and so they don't accumulate. But if that's the case, why is evolution not slowed down? And I think the answer has to do with constraint and deconstraint. That is, constraint doesn't imply that things are hard to change. It implies that there may be parts which, by virtue of their constraint, allow deconstraint around them. And these are examples in human examples where, for example, all these things are highly constrained. The caliber of screws, the electric outlet plugs, the keyboards, the integrated circuits, Lego blocks, couplers between railroad cars. All these things are highly constrained, and yet their constraint enables a tremendous amount of variation around them. You can write any book on one of these computers, and you can plug in any kind of electrical devices that were never even dreamed of at the time that these electric plugs were first invented. And you could make lots of things out of Lego blocks. So, and yet it wouldn't work unless these were highly constrained themselves. So the thing I want to focus on is the nature of these very constrained mechanisms and how they provide deconstraint for building the phenotype around them. So I'll call these things the core processes of development, and they're conserved, and we know that, of course, because many of us use this property of conservation in study of cell developmental biology and molecular biology. They have special properties. Though they're constrained, they deconstrain changes around them. And the changes involve the deployment of these core processes in different amounts, at different times, in different places, different circumstances, different orientations. Now, regulatory invention, that is modification, as we heard yesterday from Naber Barkai in the case of the promoters in yeast, is very interesting. But I think for me what's most interesting is what makes it possible to deploy these processes in different times, places, circumstances, and amounts without causing a mess. Because if you try to do that, if you take a machine like a sewing machine and take it apart into its little parts and try to reassemble it into something else, it just won't work. So there's something very special about, you can't build an airplane from a sewing machine or even anything else really, much from a sewing machine. Those parts are not meant to be reutilizable in new circumstances. Okay, so I want to talk about the properties of these core processes that enable phenotypic variation. And this brings us to the term evolvability, which I didn't put in quotes this time, but which we recognize some people think is offensive. And I think the concern with the term evolvability is that it seems a little bit like a tautology, that things evolve, have a high evolvability if they're already evolved. And maybe that's just identifying winners as being inherently more successful, when in fact they're just the ones that won out. But it makes more sense if you divide the concept of evolvability into two components, a selection component and a variation component. So by the selection component of evolvability, I mean the fortuitous relationship of phenotype to selective conditions. So organisms may not have anticipated global warming, but if you're an organism that does well at high temperature, you happen to be right there at the right place at the right time to succeed. You can't credit the organism's construction for that. So that's this fortuitous overlap between phenotype and selective conditions. But the more important one is what I would call the variation component of evolvability, and that is a measure of the intrinsic capacity of an organism to vary. And the kind of variation I'm going to talk about is to explain this variation component of evolvability is that this kind of variation in these core processes maximizes the amount of phenotypic variation, minimizes the lethality, which is obviously important because if it's lethal, it's not inherited, and produces phenotypic variation that's most appropriate to environmental conditions. And this is the one that's maybe most difficult because I'm saying it's the most appropriate to environmental conditions the organism has never previously experienced. So what is it, how can an organism have properties that are appropriate to new conditions for which it's never had an experience for that? And if it were just a reiteration of something that already happened, it would be less impressive. So I'm going to go through 4 examples of properties of biological systems that show this variation component of evolvability, this ability to facilitate evolutionary change by facilitating phenotypic variation. And there are many examples of these, and I'll only be able to show one example just to illustrate it. But the first, something we've called weak linkage, which is something that allows for easy evolution of new input-output relationships. So again, a more familiar example has to do with the electric power grid, which allows you to plug in, as I said earlier, computers, television sets, electric lights, things that have never been invented before, can be plugged into this power grid and still operate. You don't have to modify this power grid in order to operate some new component. On the other end, you can plug in also new sources of energy that had never been thought of before. You could use, you could use wind power, you could use biomass, you could use solar power. All those can be plugged into this power grid. And this facile ability to change inputs and outputs has a biological counterpart. Now, for biological systems, weak linkage has the following property. It means that you can couple processes to each other, and generally the signal is minimally informative, whereas the response is maximally prepared to be triggered. So this is the way biological systems do it. They really have these processes which are easily triggered by rather information-poor signals. And the 2 examples that happen to have the same name but nothing to do with each other are the concept of induction. The first induction of enzymes in E. coli here, the first really demonstrated clearly by Bonneau in his dioxic growth experiments, and then induction of embryonic, of the dorsal axial structure of vertebrate embryos by Speyman and Mangold. And in both cases, the same thing really is the case. That is, in the case of the inducer for beta-galactosidase, a small molecule lactose is able, as Bonneau showed, to mobilize the entire process of transcription, translation, protein folding, making a molecule that is extraordinarily complex. And we understand very well how that is done because the weak binding of this repressor to DNA is modified only enough by the binding of the sugar lactose, and that triggers a very complex response. Today, we're so familiar with this example, it's hard to realize how surprised and impressed Minot was by the fact that something like a small molecule like lactose can turn on an entire complicated apparatus of gene expression and protein synthesis. In the case of Spemann, the Spemann and Mangold experiment in the 1920s, they showed that taking a small part of the dorsal part of a gastrula stage embryo and transplanting that on the ventral, future belly part of another recipient embryo generated 2 complete embryonic axes with a complete nervous system, somites, notochord, etc. And very little of this tissue actually participated in these structures. These were all structures that were fated to become belly ectoderm and turned out to be all this complicated apparatus. And for years, people speculated at what kind of information was being transferred. And now we actually know the answer. And it's very, very disappointing on some level. All it is a couple of inhibitors of BMP signaling. And so that the little bit of inhibition of BMP signaling engenders the generation of the entire axial structure, nervous system of the vertebrate embryo. And we now know, again, that's because the potential for making all these structures exists all the way around the embryo, and it has to be, it's only elicited by a very weak inhibition, which then allows this thing to naturally occur. If such signals, like BMP inhibitors or lactose, these things can be moved around, changed in amount, and generate kind of variation in these kind of outputs. Maybe I will skip this, but just, well, I'll say quickly, you could think of a neuron as another example of weak linkage, because basically you have a system which exists in 2 states, a resting state and a firing state. The firing state and the resting state differ by the voltage across the membrane, and the firing state is a state that has unregulated secretion going on at the nerve terminal. And you can put in all sorts of ion channels and receptors anywhere in this cell and make the firing of this neuron contingent on those receptors. And there's no engineering that has to take place because there's no physical contact between the input here and the output here. The output here could be a meter away in the case of the sciatic nerve. So this is a tremendous example of the power of weak linkage when there's not even any physical linkage at all. And that enables all sorts of innovations in the nervous system to occur in evolution without having to change the basic machinery. So just to say, these kinds of processes are often self-inhibited. They're released by simple signals. They tie complex responses to simple inputs, and they can be conveyed by weak interactions. The second example is what I would call exploratory processes. And these are processes which are quite prevalent in biology, but they're usually the explanation of last resort. So people usually come upon them after having tried out other ideas. And these are processes that generate a state, first by generating many states, and then selecting among them for function. And the generation of variation may be very complicated, selection may be relatively simple. And the important thing about this for evolution is that most of the states that are generated have no use, but they can take on new uses in evolution. And these some examples of these, of course: adaptive immunity, cytoskeleton, synapse elimination, angiogenesis, and foraging, just on various levels, middle crest migration and differentiation. In all these cases, they explore the environment and selection occurs. The example closest to my heart are microtubules, which are structural elements in this cytoplasm. And these structural elements, when discovered after several years, were extraordinarily dynamic, that in a cell they were growing and shrinking all the time. which didn't make any sense, but it does make sense when you think about changing the orientation of these structures as might occur in a neurite outgrowth or some other differentiation process. And then if you have a signal here, that signal can really stabilize the microtubules that come in that vicinity. And so you don't have to re-engineer something to generate polarity. All you have to do is stabilize this dynamic array. And this kind of process can be used in many, many different circumstances. For example, the signal's here, it works. If the signal's over here, it works. Doesn't matter where the signal is, the same process will work. And for that reason, the cytoskeleton, the same basic process can underlie the differentiation of all eukaryotic cell types in which it operates, which is most. Just to give you an example of how this is used in evolution. We think about the evolution of vertebrate appendages, and we come back to Darwin's concern about how would it be possible to generate evolution of something that requires many simultaneous innovations. And so we consider about the vertebrate limb, let's say. You have to put, of course, the bones in the right place, but you also have to put the muscles in the right place. You have to put the nerves in the right place. You have to put the vascular system in the right place. Lacking any of those things, you don't have a functional limb. Lacking a functional limb, how can you select for innovation in a functional limb? But we now know from the developmental biology of these processes that once the bones are put in the right place, let's forget about that for a moment, that the muscles grow out from the flank and take their position relative to the bones, no matter where the bones are placed. You can ectopically transplant a limb bud to the head of a chicken, and muscles will migrate in, and so it doesn't require any special instruction. The nerve cells are also generated in superfluous numbers. They grow out and they innervate the muscles. And there's synapse elimination, and there's also nerve cells that don't grow and meet the muscles. They die out. So there's selection also on the nerve cell, the growth. And the vascular system is a vast system for seeking out anoxic conditions and growing toward anoxic environments. And this enables the vascular system to accommodate for weight gain, for growth, for pregnancy, for tumors, all these things, because in fact you don't need signals in this highly exploratory system. So it looks like a difficult process to achieve. Because of the nature of these exploratory processes, it becomes something that requires no new mutational change. Now there may be mutational changes that occur subsequently to stabilize these things or to make them more efficient, but to get something that you can select upon, you don't need to change things. I think I will not go through compartmentation, except to say, we'll probably hear things about this, that the compartmentation of the embryo allows the same genes to be used in different amounts, different circumstances, by virtue of selector genes that are generated in various compartments. This is sort of a schema of a Drosophila embryo. And with that, you can avoid the kind of pleiotropic problems of having to have genes that will be optimal, let's say, for one part and cause damage in another part. You can just determine their usage in specific locations. Okay, so well, just an example of that maybe would be if you transplant, if you look at the thoracic vertebrae and the cervical vertebrae, the thoracic vertebrae differ by having ribs associated with them. If you transplant thoracic vertebrae to the neck, they'll generate ribs. And if you look at the osteoblasts that are generating these structures, you really can't tell the difference between a cervical osteoblast and a thoracic osteoblast. But they are different. They are different because they come from different compartments, and they have slightly different signals. And these signals endow them with slightly different properties to respond to the signals around them, to their extent of growth. And this way, by compartmentalizing these things and having special rules for each cell in each compartment, you can use the same basic processes over and over again to generate many different types. The last property I want to mention was state selection. I don't know how I'm doing for time here. 5 minutes? Okay. And mainly what I want to say is, and this again comes back to things that were said yesterday by Nama about the sort of relationship between physiological adaptability, which there is selection for, and evolutionary adaptability. So many different systems have been, are generated so as they can vary with environmental conditions. And when you select for such a system, when you evolve such systems, you're not just selecting for the mean of its behavior, but also for the variance in its behavior. And these physiological systems are well buffered, and selection on them tends to be non-lethal because they're obviously varying within the life of an organism. The example I wanted to choose was hemoglobin here, which has different phenotypes depending on whether it has different oxygen binding curves, depending on whether you're at the top of a mountain and your body makes diphosphoglycerate, or whether you're exerting your muscles and the pH drops and you make a lot of lactic acid. That changes the phenotype of hemoglobin. And so I would say that, and this has been thought of a lot, and I want to say one thing important about this with regards to development and evolution, but I would say is that in the case of systems which are physiologically variable, you can stabilize that variability in different states and take advantage of the fact that the organism has already evolved variation which can be useful in evolution. So this is a goose that flies over the Andes at 9,200 meters, and it has a single proline replacement on the alpha chain of hemoglobin. And so it really, what it does is it moves the existing saturation of curve to hemoglobin to higher affinity to enable this goose to breathe more easily as it flies over, as compared to its conventional cousin, which lives at lower altitudes. So it didn't have to invent something radically new. It just merely had to stabilize an existing property. Now, there are a number of people who thought about phenotypic variation and physiological variation in evolution, but all this was sort of pre-molecular thinking. And I think our molecular thinking tells us that this idea of state selection is most important when it's directed toward the internal developmental processes. That is, if you consider what a cell can do, a cell can change its gene expression, it can change its degree of proliferation, it can move or not move, and the envelope of possibilities which cells already possess to be moved in one direction or another direction can enable a tremendous amount of evolution without new invention, or with just slight modification, as it was, for example, in the high-flying goose that we talked about. So, and I think that the people who are thinking about physiological variation were focused mostly on external factors, but if we think of development as really a physiological system, that is development, we have a single genome, and we're using it in different circumstances depending on signals that are coming in and out. The signals are made by other cells. This allows a tremendous amount of variation to occur by just changing gene expression, proliferation, cell movement, secretion, other factors. All right, so these are the kinds of things which I think enable, facilitate variation, make variation tend to be non-lethal. And the question always arises, why would you select for something that facilitates evolutionary variation? And evolutionary biologists are absolutely allergic to the notion that you select for future benefit. It's not fair to say we're going to hold on to this in the Cretaceous because it's going to come in handy in the Pleistocene. This is not considered kosher to do that. So as a result, we have to think about current benefits. Well, the reasons why facilitated variation can be selected, well, one reason could be as a byproduct for selection for effective development in physiology. And I gave you examples with the microtubules, with the limb development, with a variety of other things, where that kind of process, exploratory processes, weak linkage, are being used just to get effective development in physiology. And the value it placed in evolution can be thought of as a byproduct of selection for that. It could be a byproduct of selection for versatility of use. We have multiple cell types in our huge organism in our body, and they're selected for on the same genes, so you have versatile use. Once you have a process that can specify each of the 5,000 bristles on a Drosophila, how difficult can it be to specify a new bristle on a slightly new related insect? You have already evolved those processes. The more tendentious arguments are the population level. By buffering evolutionary change, you increase genotypic variation. Schmalhausen and Waddington thought about this a lot, and more recently, Susan Lindquist. And I will leave the last one out for the moment so people want to ask about. Okay. Can we go back now to what Gould said? Is phenotypic variation copious, or is it limited? Does it require extensive unselected variation? Is it in all directions, or is it preferred along certain paths? How creative does mutation have to be? And I will skip the Darwin finches, except to say that recent work has shown that only modification of 2 genes in the Darwin's finches' beaks can explain all the variation that's observed, at least in the Darwin finches. And the thing I liked the most about it was when this was repeated in experimentally by Cliff Tabin by adding BMP to the head of a chicken where he was able to generate a broader beak. It wasn't just that the beak got broad, but it fit properly into the head and the musculature. So these modifications don't require many subsequent modifications. The system is set up in such a way that it responds to change to give you physiologically normal variation. Okay, well, okay, let me just summarize then. Genetic change is required for evolution, but it may not always be the most creative part. Development channels genetic variation consistent with previous selections. Weak linkage, exploration, compartmentation, state selection increase the frequency of useful variation and decrease the lethality of variation. These constrained core processes, which have changed very little in some cases in billions of years or hundreds of billions of years, deconstrain change around them and make variation easier. And therefore, the requirement for genetic change to be especially innovative, or especially large amounts of it, or requiring many simultaneous genetic innovations is therefore reduced. So as we look on the postmodern synthesis, we have evolution that Darwin is responsible for in our modern theory of it, built on selection, which was his great contribution, built on genetic variation, which he knew had to exist, but which really is Mendel's contribution, and built on phenotypic variation, which we now understand and is going to be no one's particular contribution because it's going to be the thousands of scientists who have bit by bit put this together. And I just want to thank my co-author, John Gerhardt, whom I've discussed these ideas with for many, many years, worked with many, many years. We summarized these ideas in a book called Plausibility of Life, which I guess was on sale here for a few hours. And I want to thank you very much.
Mark, you used the argument
Yes.
that phenotypic variation builds upon what already exists,
Right.
while it was implicit that genotypic variation doesn't.
Yes.
But doesn't it?
Yes, yes, yes. No, I meant, yeah, no, it's in that respect, the outcome of genotypic variation is, you know, is obviously depends on the previous genotypic variation, but you can get the previous genotype. But that's not where the selection is occurring. The selection is occurring on the phenotype. So it was really that distinction I was trying to draw. But you're right, that that it is true, the kind of genotypic variation does depend on the structure of the genome, and it also depends a lot on the phenotype, because essentially all the replication enzymes, repair enzymes, you know, all of these processes are also part of the phenotype, and they also affect the nature of the genotypic variation. So I think that's a good amendment to what I said, or correction to what I said.
Is there some evidence for the ability to generate mutations or to evolve on a small scale, let's say to optimize the ability to generate mutation in higher organisms other than, let's say, viruses where these things happen?
To use, you mean to regulate mutational rate?
Yeah.
Well, I mean, there are always, you know, people have argued that people have argued that in microorganisms that the level of mutation is regulated by the environmental stress. But nobody has really demonstrated clearly whether you can focus that genetic change to the responsive element you would want to change. So it becomes just a general elevation. I don't know in higher organisms where that's been demonstrated, that even that elevation. I think most people think that the error rates in DNA replication are sort of set by some sort of optimization, that it is possible to make things more accurate, but at such great cost that the organism has sort of lived with that. And it's still, I think, an open question in some people's minds as to whether there are going to be some ways to tune genetic variation.
I wonder whether it's really true that selection only acts on the phenotype. Wouldn't also the development for itself maybe something which can be selected? That means, for instance, development can go fast or can slow, and in a species or an individual which does it faster than it's the other one, may be an advantage because it can reach maturity earlier.
Right. I think the development is the phenotype as much as, I mean, I think there's a kind of a fixation we have of the phenotype is the adult phenotype. But the selection is occurring on the whole life cycle of the organism. And we know, for example, there's a tremendous amount of evolutionary change that's taken place in eggs, for example, that have to do with their protection and their provision for nutrients and all these sorts of things. So, yeah, development is being selected on for two reasons. One is for the processes, as you mentioned, the speed, the niche in which the larva exists or the embryo exists, and also for the contribution it makes to the adult phenotype. But I don't think, when I talk about phenotypic variation, I'm distinguishing it from genotypic variation. I'm not talking about just the adult morphology.
The concept of a weak linkage,
yes.
The way I understand it is almost like a gain function. When you have a signal which may be weak, which activate an array of changes which seemingly are unrelated. Is it possible that natural selection selected the gain function itself?
Right.
Take, for example, an instinct, which is a repertoire of complex behavior that may be triggered by a banal signal that activate a huge amount of the brain.
Yeah. Well, I think that's, you know, I think that I was going to give an example. I mean, I don't know, I didn't, I give misjudge the time a little bit, but one example I was going to talk about was some computer modeling that Uri Alan and Kirschner did in trying to ask the question, is there some imperative here for this kind of structure of weak linkage, for example, that it comes out of something abstract, really, and not the biological features. And so there was not a gain function, but there was a question of how do you build something so it can change readily when the circumstances change, when you change the cue or the output that you want, or the input that you want. And to summarize quickly, they found that if a system, if this was a logical, it was a kind of a logic circuit that they built, or logic circuits that they built, if they built them and they kept selecting them under variable conditions, and if it had a certain modularity to begin with, then it was much more rapid in achieving new functional outputs. So it gets to the question of what is designed for effective function, and so if you're building something which can be variable, then it also can achieve, it builds a certain structure into the organism, which then allows it to evolve more quickly. So it may very well be that you were saying about instinct or about signals in the organism. In order to build an organism, if you really understood it, you'd say it would have a certain property, and that property itself would also allow it to evolve more easily. And I think I haven't, nobody I think has gotten to that depth yet, but I think those efforts of Alan and also other people in different ways to try to imagine the connection between effective function and effective evolution is kind of also what I was trying to get at in this talk.